Okay. Okay, welcome everyone. So my name is Caitlin. I am one of the facilitators for the Warren County / Front Royal Lyme and Tick Disease Support Group. And I'm joined by my colleague Jody. She is another one of the facilitators of our group. And then we also have the pleasure of hosting a guest speaker, Dr. David Gaines, this evening. So he's with the Virginia Health Department, the Office of Epidemiology. So he's going to be speaking to us this evening about ticks and tick-borne diseases in Virginia. So I will turn the mic over to him.
Okay, can everybody hear me?
Yeah.
Okay, all right. Well, today I'm going to talk about ticks and tick-borne diseases of Virginia, and we can move on to the next slide. Okay, among the 16 different species of ticks that are native to Virginia, only 3 species commonly bite people. Probably the most common cause of tick bites in Virginia is the Lone Star tick. They are a very aggressive human biter, and they are abundant throughout the eastern part of the state, all the way up to the foot of the mountains. Most of the bites caused by Lone Star ticks are caused by the tiny nymph stage ticks, but adults also bite people and they also carry diseases. And so do the larvae. They bite people, particularly during August season. They're very tiny. They're so small, they'll go through the weave in your socks and bite your ankles up. And people think that they've been bitten by chiggers. They get a bunch of itchy bites. That's Lone Star tick larvae. The next most common cause of tick bites in Virginia is the black-legged tick. Again, most of the bites to people by this tick are the tiny little nymph stage tick shown in the inset. Most people never notice this tick when it's biting them. They get bitten by the tick and they never know they've been bitten by a tick. Adult stage ticks also do bite people during the fall and late fall and winter and early spring months. And adult stage ticks have higher infection rates of the various diseases that black-legged ticks carry. But nymphs often get away with their bite because they're so difficult to see, and whereas the adults are fairly easily seen and removed before they can transmit. Then finally, we have the adult, the American dog tick. Only the adults of that species bite people, and they don't bite people very often, but this year has been a bit of an exception. I know people who've been bitten by several of them this year. Moving on to the next slide. All right. I'm going to start with the Lone Star tick. It carries can carry a lot of different disease agents. The most important of being ehrlichiosis, which can either be caused by Ehrlichia chafiensis or Ehrlichia ewingii. Ehrlichia chafiensis is more common in Virginia than Ehrlichia ewingii, and it's also more likely to cause a fatality than Ehrlichia ewingii. Lone Star ticks are also responsible for the acquired red meat allergy. This is probably the second most common cause of illness, or maybe the first most common cause of illness by Lone Star ticks, but it doesn't often hospitalize people. STAR-I, which is an acronym for Southern Tick-Associated Rash Illness, is also caused by Lone Star tick bites. STAR-I is most likely caused by an immune response to Lone Star tick saliva. Their saliva is very allergenic, and their bites almost always itch, and it's probably the allergenicity of their saliva that causes the acquired red meat allergy above. Rickettsia parkeri is another disease carried by these ticks. It's a spotted fever group agent, and it occasionally pops up in Lone Star ticks. Rocky Mountain spotted fever is another important disease that these ticks can carry. Fortunately, they are not frequently infected with it, but it can be a severe and fatal disease. Tularemia is also carried by this tick, and we see a number of tularemia cases every year. that may be associated with Lone Star tick bites. And then finally, there's the Heartland virus disease. We've never seen this in Virginia yet, but cases have been reported in neighboring Tennessee. Next. All right, Lone Star ticks, as I've said, are by far the most common tick to bite people in most parts of Virginia, especially places below 1,600 feet elevation. The nymph stage ticks are the main cause of tick bites. Their disease transmission occurs mostly during the period from spring through summer, or May through mid-August. Adult female and male ticks also bite and transmit diseases from late winter to early summer. And the tiny larval ticks can also transmit disease from mid to late summer. Lone Star ticks are mostly found in leaf litter on the forest floor or in partly shaded grass, but are sometimes found in open grassy fields next to woods. Next. Black-legged ticks are known primarily for their transmission of Lyme disease. The next most common disease they may transmit in Virginia is Anaplasmosis. Another common one that they may transmit in Virginia, but which has probably gone mostly unrecognized because most of the medical community is unaware of it, is a Borrelia Miyamoto disease. Then there is Babesiosis, which can be a serious disease, and we're starting to see it in eastern and northern parts of Virginia. And then there's the Tick-Borne Encephalitis virus, which is a severe illness which can cause fatalities. And as I've said, the main vectors of disease to people are the nymph stage during the late spring and early summer. Adult ticks do bite people much less often, but they have higher infection rates and can bite and transmit diseases on warm days of the fall, winter, and early spring. Next. Black-legged ticks occur predominantly in forest leaf litter or shaded vegetation on the forest floor, and they are rarely found in open fields or in grass unless it is at least partially shaded by trees. I'm not sure why this black-legged tick slide got mixed up. It should have followed the first black-legged tick slide, but anyway, bear with me. The nymph stage black-legged ticks are very small, move so slowly that they're easily mistaken for a tiny black speck of dirt by the persons they are crawling on. And the use of a magnification lens may be needed to tell whether it is a speck of dirt or a tick. The bites of nymph stage black-legged ticks rarely itch, so the people they have bitten rarely know they've been bitten until they come down with Lyme disease or some other associated disease. Next slide. Finally, the American dog tick. This tick is a large tick. It is most famously associated with Rocky Mountain spotted fever. However, I'm not really sure how important it is as a Rocky Mountain spotted fever vector. I'm sure it plays some role, but probably not as great a role as has been supposed. Rocky Mountain spotted fever is a very important and historic disease, and it does cause fatalities. Then these ticks can also transmit Rickettsia parkeri spotted fever, and they can also transmit tularemia, but most tularemia transmission is probably associated more with Lone Star ticks. As I've said, American dog ticks don't frequently bite people, and so they probably are not going to get away with feeding on someone often enough to transmit a whole lot of disease. I've been playing in tick-infested habitats since I was a teenager, and the last American dog tick bite I got was in 1972 or so. Although I do have an epidemiologist working in our office here, and he lives up in the woods in northern, up near Fredericksburg, and he has actually had 3 of them try to bite him this summer. So that's kind of a record. American dog ticks occur in open fields with tall grass or vegetation, such as old fields, but are sometimes also found on leaf litter on the forest floor. Next. Now I'm going to talk about the distribution of ticks in Virginia, and first I'll talk about drag sampling for ticks because that's how we know about their distribution. The ticks that bite people typically climb onto people's feet as they walk through a tick habitat. They do not jump out of trees as some people have supposed. In fact, they rarely climb very high on vegetation. Maybe they might occasionally get waist height, maybe occasionally knee height, but mostly they are lower than that. Drag sampling also allows quantitative and comparative estimates of the tick risk from one habitat to another. So we will do drags and compare the number of ticks collected per 100 square meters covered in one type of forest as compared to another type of forest, so that high-risk habitats can be identified. Next, please. This map shows the results of drag sampling of tick populations in different regions and elevations of Virginia. And starting with the lower right-hand corner, the coastal plain of Virginia has been surveyed extensively by folks at the Old Dominion University Department of Biology. And this is just a small subsampling of all of the surveys that they've done. But as you can see from their survey work, Lone Star ticks have accounted for about 92%, 92.5% of what they've collected in this region. Black-legged ticks account for about 5%. American dog ticks almost 2%, and other tick species about 0.5%. So Lone Star ticks pretty much rule the coastal plain. However, black-legged ticks can be a little more common as you get closer to the coast, especially on the eastern shore where you have cool ocean breezes coming off of water. Black-legged ticks that we find in Virginia are a northern variant of black-legged tick, and they like Connecticut summers. They don't care for the hot Virginia summers, so they're mostly found in the cooler areas of Virginia. Now, when you go into the Piedmont and survey there, Lone Star ticks account for about 95% of what we collected. Black-legged ticks about a little more than 2%, American dogs about 2%, 2.5%, and other tick species were not found in that survey. Then in the Shenandoah Valley, which is higher in elevation, somewhere between 1,400 feet and 1,700 feet, we surveyed 2 sites there to get this data, and Lone Star ticks accounted for 44% of what we collected. Black-legged ticks accounted for a slightly higher percent at 54%. So as you're going up in elevation, black-legged ticks are more common because the summers are cooler at higher elevations, and Lone Star ticks are less common because the winters are colder at higher elevations. Then when you get to a place like the New River Valley, which is mostly above 2,000 feet elevation, Lone Star ticks account for only about 1% of what is collected. Black-legged ticks account for 98% of what is collected. American dog ticks are found there, but not in huge numbers. So when you get into the high elevations of Virginia, that's when you're really, prone to be bitten by a number of black-legged ticks. And the more ticks you're bitten by, the more likely you are to acquire Lyme disease. Next slide. All right, suburban forest environments are the places where people are most likely to encounter and be bitten by ticks. And most people who get bitten by ticks and get tick-borne diseases play or live in or near forests in suburbanized areas around towns and cities of Virginia. Adult Lone Star ticks and adult black-legged ticks feed mainly on white-tailed deer. Deer are where adult male and female ticks meet and mate, and where adult female ticks most often take a large blood meal to nourish the fertilized eggs they will lay. Deer are more abundant in suburban forests than in rural forests due to limitations on hunting in suburban environments, as well as an abundance of edible ornamental plants growing in suburban yards. Particularly, they like to go into suburban yards in the fall and winter and spring months and feast on people's flowers and hostas and tulips and whatnot. Next. White-footed mice are the primary animals that carry the agent of Lyme disease, and white-footed mice are also a common host for feeding black-legged tick larvae and nymphs. White-footed mice like to nest in and under old firewood piles, scrap lumber piles, sheds, junk piles, and old leaf piles that are commonly found along the forest edges next to suburban environments. And unlike other rodent species, a white-footed mouse that is infected with the agent of Lyme disease will remain infectious to any feeding ticks for several months. Most other animals that serve in the transmission cycle for Lyme disease only remain infected for a week or two at most. Next slide. White-tailed deer are the primary animals serving as reservoir hosts for Erythema pathogens. And deer also serve as the main hosts for feeding Lone Star tick adults and are where most adult ticks meet and mate. Deer are common in peri-domestic suburban woods because they are difficult to control in those environments. And as I've said, suburbanites tend to plant ornamental plants around their homes that deer like to eat. And as both deer and Lone Star ticks are common in peri-domestic suburban forest, Erythema pathogens are more common in the deer and the Lone Star ticks living in those environments. Next. Now I'm going to move on to Lyme disease. This graph, I'm sorry I haven't updated it to the latest data, but it goes up from 1990 to 2017. And as you can see, in the early days, there weren't many black-legged ticks in Virginia, and most of them were relegated to the eastern shore of Virginia, that they had migrated down, not migrated, their population had expanded down from New England along the coast, which is a cool environment, and onto the eastern shore. And then we started seeing cases in Northern Virginia in the early 2000s. And that's when the case numbers started to climb because they're in a rich suburban environment, lots of people around to infect. And then in 2007, Lyme disease pretty much exploded, went through the ceiling. And it has gone steadily upwards ever since then. Let's see the next slide. Okay. The map on the left shows Lyme disease cases in 2005 in Virginia. And you can see that some of those Northern Virginia counties, particularly Loudoun County and Fauquier County and Warren County, I think that's Warren County, yeah, were Lyme disease hotspots. Places like Stafford County. Fairfax County and Prince William County were also Lyme disease hotspots, but they have such huge populations that even though they had a lot more Lyme disease cases than any other counties in the state, the percentage of cases per population was lower than, let's say, more sparsely populated or suburbanized counties like Loudoun or Fauquier. And by 2005, we were starting to see Lyme disease cases in Southwest Virginia. Now, by 2015, the northern variant black-legged tick, which originally had been established on the eastern shore of Virginia and then later in northern Virginia, had spread down the Blue Ridge Mountains, down the Shenandoah valleys, both sides of the Blue Ridge Mountains, all the way to Southwest Virginia. And you can see, you know, the hot counties are still hot counties. They are heavily suburbanized areas because they're close to the major population centers of Virginia, or they are high in elevation. In the Shenandoah Valley, you see, Rockingham County and Augusta County, which have some fairly large human population centers in suburbanized areas. And then when you get into the New River Valley and the high mountains southwest of Roanoke, you're in black-legged tick paradise because summer temperatures up there are pretty much like summer temperatures in Connecticut. And the black-legged ticks can do very well down there. And there's a lot of Lyme disease transmission. Next slide, please. Now, if you look at this, this is a dot map, each dot representing one Lyme disease case in the period from 2008 through 2015. You can see Fairfax County and Alexandria and Arlington, they are just solid red. There's a lot of people there. There's a lot of suburban forests there. And so a lot of people were, Fairfax County for a long time was the leader in Virginia for the number of cases, but not for the highest infection rates because Fairfax County has a large human population. Loudoun County had much higher infection rates because slightly less human population. And then if you look at the patterns of red dots, they are mostly concentrated around major towns and cities and along roads. You may ask, why along roads? Well, it is along roads that suburban developments get built. So people put in new housing tracts along roads, and that's where you see human cases happening in the suburbanites who live next to those woods. Next slide, please. Now this eliminates, this is the same map, same data, it just eliminates the dots so you can see where the roads are, and you can see how Lyme disease seems to follow major highways and roads to a certain extent, and seems to be concentrated around heavily suburbanized areas of the state. It becomes much less common out into the rural areas of the state, and much less common as you go eastward towards the coast and into the lower elevations of Virginia, where summertime temperatures are extremely hot. But then you get to the eastern shore, then it pops up again. Places like Virginia Beach, Norfolk, the Hampton Roads, they're close to water, they get coastal breezes, and they're suburbanized, so you get cases down there. Next slide. And this shows the geographic Lyme disease incidence based on number of cases per census block group in Virginia. So it's more like a cases per population map. And you can see the area on both sides of the Blue Ridge and the heavily suburbanized areas of northern Virginia, and the areas in Southwest Virginia, particularly Floyd County, which is a very high elevation. It's 2,400 feet in most places. Those are areas that are high Lyme disease hotspots. Next slide. Now I'm going to talk about the diseases that these ticks carry and start with Lyme disease. Lyme disease is characterized by 3 stages of infection, with the early localized infection occurring 30 to 30 days after an infectious tick bite. And the most common sign of early localized infection is the bullseye rash or EM rash. But it may be accompanied by fever, headache, and muscle or joint aches and swollen glands. If not treated in this stage, Lyme disease can move to the early disseminated infection, and the symptoms of this occur anywhere from 1 to 4 months after the initial infection. And early disseminated Lyme may cause some of the following symptoms, including multiple EM rashes, which Lyme disease disseminates through the skin, so it goes to other parts of the skin and creates more EM rashes. Severe headaches, shooting pains, tingling or numbness in the extremities, Bell's palsy, cardiac heart block, and Lyme meningitis. Fortunately, most people don't suffer from those extreme forms of the early disseminated infection, but some do and get hospitalized. Then, if not treated during the early disseminated infection, Lyme may move into the late disseminated stage, which may occur anywhere from 3 months to years after the initial infection. And it may cause symptoms such as severe arthritis or pain and swelling of large joints, and the most commonly affected joints are the knees. Also, fatigue and neurological and cognitive disorders may accompany the late disseminated stage of Lyme disease. Next slide. The Erythema migrans rash or EM rash occurs in about 80% of cases, and it typically causes little sensation and may not be noticed if it is under the hair on the scalp or on a person's backside. The rash is typically 3 to 6 inches in diameter, and I believe the case definition for Lyme disease states that the rash must be a minimum of 3 inches in diameter to be counted. The EM rash may come in a variety of shapes. It doesn't always appear as a target pattern and is sometimes just a large red patch. If it is less than 2 inches in diameter, oh, okay, I was wrong, less than 2 inches in diameter, we do not count it as an EM rash. Now, there is a similar rash caused in association with Lone Star tick bites. In most cases, it looks like the kind of an angry red spot you see in the lower right-hand corner there. And that is often misdiagnosed as Lyme. So there are a fair number of Lyme disease cases being misdiagnosed, or I mean, a number of Lone Star tick bite cases that are being misdiagnosed as Lyme. The rash that results after a Lone Star tick bite is not usually anything more than an immune response to Lone Star tick saliva. It does not indicate that a disease agent has been transmitted, unless you would consider Lone Star tick saliva a disease agent. And I guess you could sort of consider it a disease agent because Lone Star tick bites do cause an acquired red meat food allergy, or anything that contains any type of food that contains alpha-gal, which is also found in Lone Star tick saliva. Next slide, please. Now, Lyme disease may be diagnosed by the presence of an Erythema migrans rash. Okay, and here I'm back to 3 inches in diameter. I'm going to have to get my facts straight. Sorry about that. This method might not be reliable because of the Lone Star tick rashes. Lyme disease is also diagnosed by the use of two-tier ELISAs and Western blot assays for IgM within 30 days of illness onset, or by uses of an IgG Western blot assay that may be positive at any time in the course of illness. But generally, it does not become positive until about 30 days after the initial infection. Next slide. Moving on, other black-legged tick-associated diseases in Virginia. Anaplasmosis is a bacterial rickettsial disease having many symptoms in common with ehrlichiosis and with Rocky Mountain spotted fever. Anaplasmosis is not as common in Virginia as ehrlichiosis. I don't know why I have the recently, excuse me. We typically only identify an average of about 13 anaplasmosis cases per year, compared with 102 ehrlichiosis cases per year. And those are annual averages from 2015 to 2020. To date, most confirmed anaplasmosis cases have occurred in northern and eastern Virginia, where black-legged ticks and Lyme disease have been established for the longest in Virginia. Anaplasmosis may eventually become much more common throughout Virginia. Next slide. Babesiosis, another black-legged tick-associated disease, is caused by a protozoan parasite called Babesia microti. And like anaplasmosis, babesiosis was slow to follow the black-legged tick into Virginia. And to date, most identified cases have been in the northern and eastern sections of Virginia, where black-legged tick has been present the longest. It is kind of strange, but some of these diseases just do not show up as soon as the black-legged tick shows up. It takes their establishment for 10 to 20 years in a place before the disease follows the tick. And I'm not sure why that is. It may be that there are other diseases that have to come into association with the tick, or other microbiology that is influencing whether the diseases can infect the tick. In any case, a babesiosis infection may cause no symptoms in infected persons, but may also result in a nonspecific flu-like symptoms such as fever, chills, myalgia, fatigue, and jaundice, secondary to hemolytic anemia, which may last from several days to several months. Dual infections of both Lyme disease and Babesia microti can increase the severity of both diseases. Next slide, please. Babesiosis infections can be diagnosed by the use of light microscopy to identify the parasites in whole blood, or by use of molecular PCR testing of whole blood samples, or by a serological assay. Next slide. Other black-legged ticks associated diseases in Virginia are tick-borne encephalitis virus, or TBEV, which is caused by a flavivirus that is distantly related to the mosquito-borne West Nile virus or to the St. Louis encephalitis virus, but is much more closely related to the Powassan virus, which is a tick-borne virus that occurs in the northern Midwest states and Canada. The tick-borne encephalitis virus is transmitted to people by bites of the black-legged tick. And to date, there have only been 2 cases of tick-borne encephalitis virus identified to have been acquired in Virginia. One of these cases was a resident of Northern Virginia who was infected while deer hunting in Warren County. Surprise, surprise. The other case was in an out-of-state visitor infected while hiking on the Blue Ridge Parkway, or near the Blue Ridge Parkway. Next slide. The tick-borne encephalitis infection in the Virginia resident resulted in long-term neurological deficits or impairments. The tick-borne encephalitis virus infection in the out-of-state visitor resulted in severe neurological impairments and ultimately resulted in the death of the patient. Both of Virginia's tick-borne encephalitis virus infections resulted from bites by adult-stage black-legged ticks in the late fall season. Both cases occurred in November. But tick-borne encephalitis virus transmission may occur at any time of the year that black-legged ticks are biting, from late September through mid-July, and may be transmitted by all stages, the adult stage ticks, the nymph stage ticks, and the larval stage ticks. Diagnostic testing by serology may be cross-reactive with the testing for West Nile virus or St. Louis encephalitis virus. But specific testing can be performed by tick-borne encephalitis virus by the CDC Division of Vector-Borne Infectious Disease. And that's where we have sent the samples from the 2 patients that were infected in Virginia. Next slide. A new tick-borne illness has been detected in Virginia, and it's known as Borrelia Miyamoto disease. Borrelia Miyamoto is a spirochete bacteria that belongs to the tick-borne relapsing fever group of Borrelia. It is in the same genus as Lyme disease, but is only distantly related to it and is not serologically cross-reactive on the test for Lyme. It is transmitted by Ixodes scapularis, the black-legged tick. Now, Borrelia Miyamoto disease cases, even though it's not reported in Virginia, have already been identified in patients as far southwest as Roanoke, Virginia, and are likely to occur in all regions of Virginia that have black-legged ticks and Lyme disease transmission. Next slide. Most patients that have Borrelia Miyamoto disease will have a fever, chills, and a severe headache, but might also have body aches, joint pains, fatigue, and nausea. Less common symptoms may include a rash, labored breathing, abdominal pain, and a lack of appetite. A fever may be relapsing in some individuals. Fever episodes may last for 2 to 4 days with intervals of 2 days to weeks between fevers. Borrelia Miyamoto has been known to cause a severe neurological illness such as meningoencephalitis in some patients. And in a 2013 to 2014 study of 97 PCR-confirmed Borrelia Miyamoto disease cases in New England, 24% of the patients were hospitalized. So it can be a severe disease. It is not reportable in Virginia yet, and I'm afraid that many docs are unfamiliar with it and would not think to test for it. It would be great to get the word out to the docs, the population of healthcare providers out there, that this is something they should be on the lookout for, because it has been found all the way as far south as Roanoke. Next slide, please. Next slide, please. Oh, okay. Now I'm going to talk about rickettsial diseases. And first I'm going to review the differences among Rocky Mountain spotted fever, ehrlichiosis, anaplasmosis, and rickettsia parkeri. All of these diseases, anaplasmosis is the only rickettsial disease associated with black-legged ticks. Rocky Mountain spotted fever and ehrlichiosis are associated with Lone Star ticks. Rocky Mountain spotted fever may also be associated with American dog ticks. And rickettsia parkeri is associated with both Lone Star ticks and American dog ticks. Typically, a patient with Rocky Mountain spotted fever will have a rash. About 90% of Rocky Mountain spotted fever patients develop a rash. And that rash typically starts on the wrists and ankles and spreads to the rest of the body. The rash is a petechial rash because Rocky Mountain spotted fever, one of the things that this disease agent does is damage the vascular system and causes vascular leakage, which causes most apparent damage at constrictions in the body, such as the wrists and the ankles. And that's where blood starts leaking out and causing petechial rash on the ankles and wrists. But this is also followed by rash spots. This is a severe illness that can cause permanent organ damage and limb amputation in survivors and is fatal in up to 25% of untreated patients and in 3% of hospitalized patients. Reason that it causes limb amputation is that it pretty much destroys the vascular system, restricting blood flow to the limbs and eventually restricting blood flow to the organs in the brain. It's a really nasty disease, and I'm happy that it's not common in Virginia. With ehrlichiosis, a rash may be present in about 60% of children and up to 30% of adults. And in serious cases, ehrlichiosis may cause neurological symptoms, bleeding disorders, or respiratory failure, or disseminated intravascular coagulopathy. And it can be fatal in up to 2.5% of hospitalized patients. The rash associated with ehrlichiosis may occur on the wrists and ankles, so it may be confused with Rocky Mountain spotted fever, but it can occur in various different parts of the body. Then with anaplasmosis, the development of a rash is rare, and anaplasmosis symptoms may be similar to those of ehrlichiosis and can be fatal in up to 0.5% of hospitalized patients. With rickettsia parkeri disease, also known as tidewater spotted fever because it was first discovered at the Portsmouth Naval Hospital in the Tidewater region of Virginia. This illness ranges from mild to severe, but is not life-threatening. A diffuse spotted rash may occur across the patient's body, and there will always be an eschar at the tick bite site. Next slide, please. The top picture shows a typical Rocky Mountain spotted fever rash, which starts on the wrists and ankles and becomes petechial. Ehrlichiosis rashes, as I've said, are variable, but might also sometimes appear on the wrists and ankles and can also cause petechiae. And then down below, you will see on the left a person with a rickettsia parkeri rash, widely separated spots on the body. The most distinctive thing about rickettsia parkeri infections is that you will develop an eschar at the site of the tick bite. This starts out as kind of a pimple, pus-filled pimple that eventually ruptures and festers, starts to look like a small volcano. And in fact, it is an ideal spot to collect diagnostic samples for sending to the CDC. The scab of the eschar or any of the fluids inside the eschar will be full of Rickettsia parkeri bacteria, which can be detected by PCR testing. But unfortunately, the only people who do that are at the CDC, rickettsial zoonosis branch. Next slide, please. Fortunately, although Rocky Mountain spotted fever is frequently diagnosed by doctors in Virginia, it appears to be rather rare. We typically count less than 1 confirmed cases per year in Virginia. This year is a little bit unusual in that we have counted 2 cases, whereas in most years we don't count any confirmed cases. The overdiagnosis of Rocky Mountain spotted fever is due to the fact that a large proportion of the Lone Star ticks in Virginia carry a spotted fever group agent called Rickettsia amblyommatis. Most persons exposed to Lone Star tick bites in Virginia are exposed to Rickettsia amblyommatis. And although this agent is not known to cause any significant illness in people who are infected by it, exposure causes all exposed persons to test positive on the test for Rocky Mountain spotted fever for up to a year or more after their Lone Star tick bite. And there are some people in Virginia that live in the countryside or in the suburbs that do spend a lot of time in the woods and don't take tick precautions. They get bitten by Lone Star ticks on an annual basis, and they may just continually test positive for Rocky Mountain spotted fever for years on end. And as many people get Lone Star tick bites, oh, I've covered that. All right, next slide, please. We generally see less than 1 confirmed Rocky Mountain spotted fever case per year, compared to approximately 100 ehrlichiosis cases, with about 40% of those ehrlichiosis cases confirmed. Tick surveys have shown that the agents of ehrlichiosis are much more common in Virginia's ticks than the agent of Rocky Mountain spotted fever. The severe symptoms of ehrlichiosis can be mistaken for the symptoms of Rocky Mountain spotted fever, but is much more likely for Virginia residents with a severe illness to be suffering from ehrlichiosis than Rocky Mountain spotted fever, even if a doctor's initial serological test results are positive for a Rocky Mountain spotted fever infection. Now, a lot of docs, they see a patient that has symptoms that might be similar to Rocky Mountain spotted fever, and the first thing they do is they draw blood, draw serum on that patient, and send it off for Rocky Mountain spotted fever testing. And a pretty high percentage of those serum samples test positive for Rocky Mountain spotted fever. And that is because of the non-pathogenic Rickettsia species carried by Lone Star ticks. And unfortunately, I mean, most of these patients, they get treated. Probably most of them, if they are ill, are really infected with ehrlichiosis. So their cases are not counted as ehrlichiosis. So I suspect that we are greatly undercounting ehrlichiosis cases in Virginia and overcounting Rocky Mountain spotted fever cases. Recently, the national case definition for Rocky Mountain spotted fever was changed, and the titer required to consider a case as Rocky Mountain spotted fever was raised to 1 to 128 instead of 1 to 64, just because a lot of people get tested for Rocky Mountain spotted fever in the first days of illness, when they should not even be testing positive, because it takes at least 10 days for a person to develop a detectable immune response to Rocky Mountain spotted fever after their onset of symptoms. And most people see their doc in the first day or two after onset of symptoms. That's when they test positive for Rocky Mountain spotted fever. But that's an indication that what the doc is detecting is an old exposure to a Lone Star tick bite. Moving to the next slide. This graph shows the incidence of Rocky Mountain spotted fever, ehrlichiosis, and anaplasmosis in Virginia from 1990 to 2015. And eventually I'm going to have to update my graphs, but I've been busy with other things these days. From this graph, you would get the impression that Rocky Mountain spotted fever was really common in Virginia, and as I've said, it is not. If you look at the other lines on the graph, particularly for ehrlichiosis and ehrlichiosis anaplasmosis unspecified, some of those bumps, peaks coincide with the peaks in the blue dashed line, suggesting that, well, these are related, related to Lone Star tick bites. And probably the curve for ehrlichiosis should be much higher. Next slide, please. This map shows the ehrlichiosis and anaplasmosis cases identified by serologic assays in Virginia from 2006 to 2015. And as you can see, ehrlichiosis really accounts for the majority of cases, but there are a fair number of anaplasmosis cases in there. However, serology for ehrlichiosis and anaplasmosis is cross-reactive. So a person with ehrlichiosis can be tested for anaplasmosis and will show up positive and have almost the same titer as a person who has ehrlichiosis and is tested for ehrlichiosis. Then there are also these cases of ehrlichiosis, anaplasmosis unspecified. Those are people who tested with equal titers to both diseases, and we just couldn't even guess at what the cause of the illness was. And you can see a lot of these cases are concentrated in heavily populated Northern Virginia, Richmond City area, the Charlottesville area, the Fredericksburg area, Petersburg, Williamsburg, and other major population centers of Virginia, showing the influence of suburbanization. Next slide.
Dr. Gaines, I'm sorry to interrupt you, but real quick, I just want to do a time check. It's about 7:48. So we want to leave some room for some time for questions at the end. Okay. So I don't know how many more slides you have, but just a little time check for you.
I can finish after the next, I think, 3 or 4 slides.
Perfect. Okay.
Okay, next slide. Now this graph shows ehrlichiosis and anaplasmosis cases identified by multiplex PCR assays from 2006 to 2015. And as you can see, Ehrlichia chauffiensis pretty much dominates. There are some Ehrlichia ewingii cases, mostly in the Warrenton and Fredericksburg area. And as you can see, anaplasmosis cases are mostly in Northern Virginia, a few cases in the coastal Tidewater area, 1 case in Lynchburg and 1 case in Blacksburg. But really, ehrlichiosis rules. Now, you may wonder, why is Ehrlichia ewingii absent in all these other places? Well, that's because those people were tested by multiplex PCR by LabCorp. And LabCorp's multiplex PCR test does not test for Ehrlichia ewingii. The yellow dots around Fredericksburg and the Warrenton area, those people were tested at the hospitals in Fredericksburg and Warrenton, which do their testing through the Mayo Clinic lab. And the Mayo Clinic lab's multiplex PCR tests for Ehrlichia ewingii. So there are probably a lot of people who contract Ehrlichia ewingii in Virginia and are said, well, you don't have ehrlichiosis because they were tested at LabCorp which doesn't detect Ehrlichia uniae. Just a note. That's my pet peeve with LabCorp, just one of the many. Moving on to the next slide. Okay. As you can see, these maps show spotted fever rickettsiosis cases, ehrlichiosis cases, and anaplasmosis cases. And the spotted fever rickettsiosis cases and ehrlichiosis cases pretty much cover the same areas of the map. And it's likely that a lot of those cases are ehrlichiosis. That's the only thing I wanted to show from this slide. Next slide. Okay, I think I'm done. That is, I could go on about tick prevention, but if you would rather ask questions, I'm happy to answer at this point.
Yeah, I think we'll open the floor to questions. Everybody is more than welcome to send them in the little chat bar. It looks like we may already have a question there. Is that a question? What is that?
Oh, I think that was from Clarissa before. She was saying 5 centimeters or greater for EM per our case definition. Oh, I think that was from one of the earlier slides.
Okay. So Clarissa verified that it's 5 centimeters or greater. What does it say for EM?
So that's about 2 inches.
Okay, there you go.
Sorry for the error in my slide saying 3.
No worries. So yeah, if you have any questions, just go ahead, send them in. And we have Clarissa, who is a epidemiologist for the Lord Fairfax district, here on the call as well, who can answer any questions that might be more local. But, you know, of course, Dr. Gaines is also a wealth of knowledge.
I have a question while we're waiting. Do you ever find Babesia duncani? You talk about Babesia microti.
Babesia duncani is, you know, some of the Lyme literate docs in Virginia test for that, but it is a Western Babesia species that does not occur in the Eastern United States. And so possibly if they get a positive, it's reacting with either some non-pathogenic Babesia species or with someone who has one of our local Babesias, although you would think the local person would test for all the different Babesia species if they were testing for duncani.
Okay.
I chalk them up as false positives.
I see. So just, it's a Babesia, just it's cross-reacting with a different type of Babesia.
Yeah, and it is a West Coast species. It's not found anywhere east of the Sierra Mountains.
Yeah, I had read that, but I thought, well, maybe they're migrating this way. Or someone. Okay.
Okay. Well,
I think we do this way.
okay.
Any ideas on how to help physicians be more aware of what to test for in one area versus another?
Well, I would say, you know, certainly Lyme disease, while most common in the high mountains of Virginia, it can occur pretty much anywhere in the upper Piedmont and Shenandoah Valley. And it is a pretty common disease. Most people don't ever realize they've been bitten by a black-legged tick nymph because they're so small and their bites don't itch and they can feed on you for 5 days and drop off and you'll never know they were there. So Lyme disease should always be considered if the symptoms match. I would say anaplasmosis should be considered. Certainly in Lone Star tick country, one should always consider ehrlichiosis before anaplasmosis because anaplasmosis does not appear to be extremely common in Virginia. It may become common in the future unless global warming drives the northern variant black-legged tick back to New England. It might become more common in Virginia, but really ehrlichiosis is quite common. And one of the things that docs need to do is stop using serological testing for ehrlichiosis because it cross-reacts with the anaplasmosis, and use the multiplex.
Library closes in 5 minutes. If you have items to check out, please.
Yeah, the serologic assays are not near as good as the multiplex PCR assay, and in particular, the multiplex PCR assay used by the Mayo Clinic lab. As for what else to test for, one really needs to look at the patient's symptoms, and if one can identify the tick that bit the patient, it's really hard to say what to treat a patient for without the symptoms or without knowing what tick bit the patient. But you know, certainly Lone Star ticks are one of the most common causes of tick bites in Virginia, except for parts of Virginia in the higher mountain regions.
We got one more question. Someone says to get an Ehrlich positive, do you have to be tested for that or is it correct you have to be tested for that rather than Lyme? And if you've been bitten by a Lone Star tick, do you have to request for that, or how do you convince the doctor?
Well, I wouldn't test for anything unless I was sick. I mean, if one is bitten by a Lone Star tick and one develops an onset of symptoms that may be compatible with ehrlichiosis, certainly you want to be tested for ehrlichiosis, and it's best to be tested by the multiplex PCR, because, you know, if you have Ehrlichia ewingii and they test you with the LabCorp test, the doc will say, no, you don't have ehrlichiosis. We don't know what you got, but you don't have ehrlichiosis. And you know, it's a shame that LabCorp offers a test that doesn't include that Ehrlichia, because it's probably fairly common in Virginia.
All right. Oh, and this is, thank you, Dr. Gaines. Very informative.
You're welcome.
Okay.
Yes, thank you very much. I really enjoyed that. It was informative.
Yeah, sorry for the boo-boos.
Oh,
that
no
was great.
problem.
Yeah.
No problem.
Thank
Okay.
you very much.
All right, thank you. You all have a good night.
All right. Thanks, Dr. Gaines.
Okay, thank you very much.
Bye, everyone.
Bye.